Okay, in this video, I want to talk to you about what happens to the chylomicrons and the VLDL lipoprotein particles once they are fully constructed and circulating in our bloodstream. So let's start out with the main structures, chylomicrons, VLDL. So recall that chylomicrons are synthesized by combining the triglycerides and cholesterol esters, and phospholipids around the ApoB48 structure. And the VLDL is similar manner constructed around ApoB100. These are produced, put into the blood, and then they need to be processed. And what processed means is that we need to be able to extract the fatty acids from the core structures in order for it to be delivered to the tissues for energy utilization or storage. So the chylomicrons and VLDL are traveling through the circulation, and they need to be processed in order to extract the fatty acids and deliver cholesterol to the cells. And this is done by the enzyme lipoprotein lipase. Now, as it would be, the lipoprotein lipase is produced by the cells that need the fatty acids for either energy, which would be primarily skeletal muscle, cardiac muscle, or it's produced by the storage tissue, adipose tissue, in the adipocytes themselves. So what happens is, remember the orientation we have. We have, for example, skeletal muscle on the abluminal side of the vessel. So this is the circulation over here. This particular line here represents the endothelial barrier that separates the circulation from the tissues. We have skeletal muscle in the tissues. We also have adipose in our tissues. And now the conundrum we have here is that the fat is located in here in the lumen of the vessel, and it needs to get across the endothelial barrier and into the tissues. So the question is that if you have your chylomicrons and VLDL particles that are carrying your fatty acids and cholesterol, how do the tissues here know that these particles are available to them in the circulation? Well, what happens here is that these tissues make lipoprotein lipase, which is abbreviated LPL. And the LPL is synthesized by skeletal muscle and by adipose tissue, and it's transported across the endothelial barrier by a process called transcytosis, which goes from the abluminal side to the luminal side of the endothelial cells. And this LPL is now exposed to the chylomicrons and VLDL so that it has access to do the lipase activity. Now, another consideration is that, remember, the circulation, we have an enormous amount of blood flow. We have a huge pressure of the LPL, potentially, once it reaches the circulation, of being rapidly swept away and downstream. Well, if the skeletal muscle is here and the LPL is swept way downstream, any potential lipase activity is going to be releasing fatty acids downstream from the tissue that needs it. So in order to localize this hydrolytic reaction within the tissue itself, we need to tether the LPL enzyme to the wall of the endothelial cell. And this is done by a specialized carbohydrate structure that is called heparan sulfate proteoglycan. Now this is a molecular structure that we haven't talked about yet in class. The heparan sulfate represents the carbohydrate portion of this molecule. The proteoglycan means that it is a protein with a carbohydrate modification to the protein. As it turns out, the structure of this particular heparan sulfate proteoglycan, which is abbreviated HSPG, what tethers the structure to the surface of the endothelial cell is a protein core. Off of this protein core, we have these long polysaccharide structures. And these polysaccharide structures are represented by the heparan sulfate group. The LPL enzyme has a very high affinity for binding to this heparan sulfate portion of the molecule. Now this orients this molecule, the LPL, into a position so that it binds to the HSPG, but its active site for lipolytic action, lipase activity, is exposed to allow it to interact with these potential fatty acid donors. Now in doing this, these structures flow by, and as they come in contact with the LPL, we have a hydrolytic reaction where the LPL now releases fatty acids plus glycerol. So in order to release fatty acids and glycerol, the LPLs, the lipase activity, is primarily functioning on the triglyceride structure. These fatty acids are now transported across the endothelial barrier and transported into the skeletal muscle. Now in skeletal muscle, under these conditions, are going to turn this into ultimately CO2 plus water plus ATP. Likewise, adipose is going to do the same process. It is going to also synthesize the lipoprotein lipase, transport it across the endothelial barrier, and tether it, bind it to an HSPG structure. This, again, same manner as the skeletal muscle, holds the LPL in position, which is within the blood system, circulation system of the tissue that needs the fatty acids. Now, as VLDL gets processed, it is going to continue this hydrolytic reaction to release more fatty acids from the structure. In the case of adipose, these fatty acids will be transported into the tissue itself, and the adipose tissue will reconstruct the triglyceride for storage. Now, recall about the density of VLDL. This is a very has low density, and that's because it has a very high lipid to protein ratio. Now, as the LPL, the lipase, performing its hydrolytic activity here, you will notice we're releasing lipid. So that is going to increase the density of the VLDL. After we do sufficient processing of this, we remove enough lipid so that our lipid to protein ratio drops significantly, and now it becomes an LDL particle. So this is the relationship between VLDL and LDL in the circulation. Now let's go back to the chylomicrons. This, of course, the chylomicrons are occurring in a fed state. Chylomicrons are undergoing the same hydrolytic activity by the lipase enzyme. The lipase is removing fatty acids from the triglyceride stores in the chylomicron. These fatty acids could potentially go to skeletal muscle, but remember this is the fed state. So is this a prime energy source for skeletal muscle in the fed state? Probably not, because the skeletal muscle, assuming a balanced diet, is going to have plentiful amounts of glucose. So the pathway for chylomicrons is going to be mostly through this pathway to release the fatty acids into adipose for storage as triglycerides. When a sufficient amount of this is processed, then we have our chylomicron remnant. Now, once the chylomicron remnant is formed, this becomes a spent product, as we talked about before, and the spent product is now recovered by the liver. The liver again will disassemble the chylomicron remnant and reassemble it with ApoB100 as the skeleton protein and form the VLDL particle, which is then put back into the circulation, so ensuring that cells and tissue have an adequate supply of fatty acids in the fasting state. Now, the fate of LDL has two paths. The first one is that LDL can itself pass into the tissues. Now, it can pass into the tissues, recall, because LDL is a much smaller particle, and there are specific transport mechanisms allowing the uptake and passage across the endothelial border, bringing it into the tissue. In this manner, this is a manner that we can bring cholesterol to cells. Recall that the VLDL is being processed into this lower density structure because we're losing lipid. The lipid we're losing is triglyceride. What remains largely in the LDL particle is cholesterol. So by processing VLDL, we can release fatty acids to tissues for energy, tissues for storage, and then the remaining, considered a spent particle, LDL, can be transported and deliver its balance of its lipid cargo of cholesterol to cells. The other path that LDL can take is, you guessed it, to the liver. And in this manner, the liver can take up the LDL particle and release the lipid components, remaining lipid components from the ApoB100, and the cholesterol can be used, recycled into biosynthesis to rid the body of excess unused cholesterol. So this is the path of delivery of fatty acids and cholesterol to peripheral tissues after we formed our delivery vehicles, chylomicrons and VLDL. And the next step we have to talk about is the lipoprotein uptake by cells. What is the exact mechanism that our chylomicron remnants and our LDL are being taken up? This is a receptor mediated process. In other words, there are specific cell surface receptors on tissues, and in this case the liver, that can recognize these as ligands in cargo, bind them, and bring them inside the cell. Now let's take a look at the uptake and internalization method. The first one, which is the most straightforward, which has been studied the most of all internalization receptors, is the LDL receptor. Now, recall the fact that LDL is the vehicle to deliver cholesterol to cells, so that means that all cells have an LDL receptor. So the mechanism we're about to look at is consistent with all cell types. So the LDL receptor is located on the surface of a cell. We have the N-terminal portion of the protein sticking out into the extracellular space. We have the C-terminal portion of the receptor sticking into the cytoplasm. Now, LDL particles have, as part of their core, the ApoB 100. Now, I mentioned that the ApoB 100 serves as the skeleton to form the VLDL particle by adding in the triglycerides, the phospholipids, the cholesterols, cholesterol esters. It also has a recognition function. And this ApoB portion of the LDL particle itself can bind specifically to the LDL receptor. This is a high affinity interaction, and once this occurs, we have a pinching off of the membrane, an endocytic event, as it is called, endocytosis, and this process brings the LDL to the intracellular compartment of the cell. So once this vesicle pinches off, it will form a vesicle inside the cell that is called an endosome. Inside the endosome, we have our LDL receptor across the membrane, and we have our LDL ligand bound to the receptor. Now keep in mind the orientation, because this is an internalization event. The inside of this lumen, is consistent with the outside of the cell. And so therefore we have internalized the LDL into this endosomal compartment. Now, once this endosome is brought into the cell, it begins what's called an acidification process. The acidification is simply there to drop the pH within the lumen of the endosome. In doing so, this vesicle will now be able to separate the ligand from receptor. So the LDL will now dissociate from the receptor. Here, we eventually form two vesicles. This vesicle here is called a lysosome. The pH of this lysosome is approximately 5. So this acidification process drops the pH of this vesicle by 2 orders of magnitude. In this vesicle here, we have the LDL particle. In this vesicle, we have the LDL receptor. So we have been able to separate by dissociating the binding from LDL from its receptor, by acidification, dropping the pH, causing this dissociation, and now we're separating the cargo, the LDL, from its receptor. The receptor enters a vesicle that is called a recycling endosome. And just as that name indicates, this vesicle will recycle back to the cell surface. The vesicle will fuse with the plasma membrane, and the LDL receptor is now exposed to the outside to receive the next LDL particle that needs to be endocytosed. Now the LDL that is now localized in the endosome is going to be degraded by lipases, by esterases, to hydrolyze the remaining triglyceride that may still be associated, remove the cholesterol from the particle, proteases, to break down the ApoB100, and these are all specialized hydrolytic enzymes that are called acid hydrolases, because these particular enzymes are different than the ones we've looked at before. The ones we looked at before were functional in neutral pH. These hydrolases work under acid conditions because they're in a late endosome or lysosome with a pH of 5.0. So therefore all of this cargo gets broken down, degraded, and discarded. So that's the uptake, the binding, uptake, internalization, and processing of the LDL particle. Now the chylomicron remnant also undergoes a receptor-mediated event, but it's slightly different because this is happening primarily on the liver. And we have to concern ourselves here with an anatomical structure called the space of Dissay. The space of Dissay is a space that is localized on the surface, between the surface of the hepatocyte and the circulation. So if the circulation is up here at the top of our drawing, these are endothelial cells, both of these structures. This is a pore, a gap between the endothelial cells, and then here we have the top of our hepatocyte, which is the primary site in the liver that is performing our metabolic functions. So the chylomicron remnant is in the circulation, and because of the porosity of the endothelium, the blood vessel here, it is able to freely enter the space of Dissay, which is this space occupied between the surface of the liver cell and the endothelial cell that serves as the junction of the circulation. Once the chylomicron remnant enters the space of Dissay, there are now numerous proteoglycan structures on the surface of the hepatocyte that can bind the remnants as they enter the space of Dissay. What this essentially does is it holds the remnants on the surface of the hepatocyte. Two functions this serves. First is an enzyme called hepatic lipase. Hepatic lipase is made by the hepatic cell, by the liver cell, and it serves to perform a hydrolytic reaction on the remnant while it is localized on the proteoglycan structure, which are HSPGs, just like we saw in the endothelium in the tissues of muscle and adipose. And this lipase will perform a hydrolytic reaction to release fatty acids plus glycerol, which will then be taken up into the liver cell. So that's the first function of this binding of the remnant to this HSPG. The other serves as a reservoir to hold the ligand while it's waiting for its carrier to be internalized. That carrier to be internalized is a receptor called LRP. These LRP molecules, LRP receptors, are internalized in the same manner as the LDL receptor. I'll abbreviate CR as the ligand for binding the receptor, which stands for chylomicron remnant. We undergo a dissociation event. Again, the same process of acidification, which is going to separate the receptor from the cargo of chylomicron remnant. This will ultimately be disassembled and restructured into VLDL in the liver, whereas the LRP receptor that has undergone the internalization event for chylomicron remnants will be recycled back to the surface of the hepatocyte, allowing for the next round of chylomicron remnant internalization. Now, this binding function here onto the heparan sulfate proteoglycan is essential because we only have a limited number of LRPs in order to internalize the remnant. This is a saturable process. So if we don't have this tethering here, the chylomicron remnants could get loose in the circulation and their circulation time would be prolonged, leaving a higher fat content in the body. So the chylomicron remnant comes in. If the LRP is available, it binds directly to it because it has a higher affinity than it does for the HSPG. This internalization occurs. The LRP is no longer available until it's recycled, and so more chylomicron remnants come in, bind to the lower affinity site, and are held there, being processed by the lipase, so it's an early pre-processing. When the LRP is recycled back to the surface, chylomicron remnants can now dissociate from the HSPG, bind to LRP to undergo another round of recycling.